Okay, so this is the chapter 14, part 5 video presentation. And in this section, I'm going to focus on really differences that we see in DNA replication in eukaryotes, and then just talk about some distinguishing features of eukaryotic DNA. And so here we go. So first off, we know that eukaryotic organisms, compared to prokaryotic organisms, our genomes are more complex and they're larger in size. Instead of having just one circular chromosome, we have multiple linear chromosomes. Instead of having just one origin of replication, we have multiple origins of replication. In fact, in humans, our chromosomes can have up to 100,000 origins of replication, compared to just one in prokaryotes. Even so, the rate of replication in eukaryotic organisms with all of these origins of replication is still much slower than prokaryotes. It's about 100 nucleotides per second. And so the reason for that is, not only is our genome larger, but eukaryotic DNA also has proteins associated with it called histone proteins. And in eukaryotes, those histone proteins have to be moved in order to be able to access the DNA. And so they have to be actually moved out of the way so that DNA can be replicated. We don't have that issue in prokaryotes because there are no histone proteins. So there's an added layer of complexity. What else? In eukaryotic organisms, there's more than 3 types of DNA polymerase. In fact, there's at least 14 that have been discovered so far. Okay, so more enzymes involved in this process. However, even though there are these differences, the essential steps of replication are still the same. So, the DNA gets unwound and unzipped at the origin of replication. Single-stranded binding proteins come in and bind to single-stranded DNA. A primer is still required before DNA polymerase can do its job in copying the DNA. There is still a leading and a lagging strand because DNA polymerase can still only lay down bases in the 5' to 3' direction, and because DNA is anti-parallel. Okay, so all of that is still the same. Here's something else that's a little different. So because eukaryotic chromosomes are linear, right? Bacterial chromosomes are circular, so we didn't see this issue that we're going to talk about here. Because our chromosomes are linear, when we get to the end of the lagging strand, the ends where the RNA primer comes off, they don't get copied. And so what we end up with is a hangover, well, not a hangover, DNA that's, we have more DNA on the template strand than on the new strand. Okay, and so let's look at this first picture here. Let's ignore the rest of this for just a second. We're going to look at just this. So this shows in white the original chromosome, or sorry, the original DNA, the template strand, and then in orange here is our new strand. Okay, and so again, DNA polymerase lays down the 5' to 3' direction. So it's been working in that direction. This is the lagging strand. Well, we get to the end of the DNA, and we can't add a primer. There's just not enough room here. There's not enough room for DNA polymerase to come in and fill this in. It can't. And so we end up with this overhang. So the template strand has this overhang here. And so this is missing DNA on that lagging strand. Okay. Again, we don't have that problem in prokaryotes because their DNA is circular. What can happen after rounds of cellular replication, after rounds of DNA replication, is this end of this lagging strand, these will come apart and both serve as templates in the next round of replication, which means that we can start to lose genetic information on the lagging strand. And so our chromosomes, with every round of replication, can get shorter and shorter and shorter. And what eventually happens is we lose genetic information. We lose genes. And so that can be very detrimental. And so what happens, let me go to the next slide. We're going to use the same figure first. Here we go. is our cells have evolved a mechanism to prevent this. And so there is something called a telomere. Okay, so a telomere right here is going to be a repetitive sequence that doesn't code for anything, but it gets added to the ends of chromosomes. And the reason for that is so that we don't lose valuable genetic information. So we have these ends of our chromosomes that don't code for anything. So if we lose a little bit of it, it's not a big deal. Okay, so what happens here is the enzyme, which is called telomerase. This was discovered by Elizabeth Blackburn. She received the Nobel Prize for her work in 2009. Telomerase is going to be the enzyme that adds these telomeres. And so telomerase is shown in blue in this figure. And so this is what happens. Telomerase comes in, and it has this little RNA template. And what will happen is it will, this will serve as a template. And so what happens is DNA polymerase comes in and it extends and adds a few more bases here. So we extend the template strand, which then allows enough room for here an RNA primer to get added to this lagging strand, and then DNA fills it in. Okay, so we're just extending the end of the template strand so that we don't lose the genes that could be right here. Again, look at the sequence. It's very repetitive. C, 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 A, A, U, C, 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 A, A, U, C, 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 A. That, you know, so that's, we're laying down repetitive bases here that don't code for anything. But again, the reason for that is so that we preserve coding region and we don't lose genes. So what we found, and again, this is only present in eukaryotes, not prokaryotes. We found that telomerase activity is really active in germ cells and in adult stem cells, so embryonic stem cells and adult stem cells. However, if we were to look, at our somatic cells, any of our body cells right now as adults, the activity of telomerase is low to nothing. Okay? And so what happens as we age, telomerase activity goes down, and therefore, with every round of replication, our chromosomes get shorter. Okay? And so this telomere shortening is associated with the aging process. Now, we're not sure if it's just part of the aging process or if it's actually the cause of the aging process. So if you start to lose genetic information as you age, that's a problem, right? Does that cause aging, or is it just a correlation? Is it causation or correlation? We don't know yet. Those studies are still happening. However, in mouse models, it's been shown that if you increase telomerase activity in adult somatic cells, that could actually reverse some age-related conditions. And so will that be repeatable in humans? Maybe. Is this the fountain of youth? Can we reverse aging? Yeah, maybe. Okay. We've also found this. So again, there's a link between telomerase activity and aging. We've also found this. So there's also a link between telomerase activity and cancer. And so briefly, I'll give you kind of a brief summary of what cancer is. Cancer is the result of uncontrolled cell division in abnormal cells. So these cells, something's wrong with them, and they continuously divide and divide and divide, and they don't stop. Okay? There's obviously more to cancer than just that, but that's kind of a brief summary. And so what we found is when we look inside of cancer cells is that the chromosomes inside of cancerous cells have shortened telomeres. So they've lost, ends of their chromosomes. But they also have high telomerase activity, which is really weird. And so what we think happened is telomere shortened, maybe there's even some loss of genetic information, and then the cell becomes cancerous. And at that point, as the cell's going through this rapid division, that is when telomerase activity spikes. Okay, so it was low, we've lost some DNA, and now it's become cancerous, and now it's high. Okay, so telomerase activity doesn't spike until after the telomeres have become shortened. And that's the only thing that makes sense if we look at telomerase activity in aging. Okay. So what does this mean? So again, we think we can, possibly use this to target cancer. Okay, just like maybe we can target some age-related diseases, maybe slow down or prevent aging. We think that we might be able to also target cancer cells by knowing this. So we can use targeted telomerase inhibition, so drugs that will specifically inhibit telomerase activity in these cells, and maybe if we do that, they will stop dividing and they won't be able to, to live. So that's a potential drug therapy. So again, as we talk about eukaryotic versus prokaryotic DNA replication, there's a lot of similarities. The big differences are, telomeres and telomerase are present in eukaryotes. We don't see that in prokaryotes. Eukaryotes have, many more kind of enzymes and proteins involved in the process. And the process is slower, much slower in eukaryotes compared to prokaryotes. And that's because our genomes are larger and we also have histone proteins. And lastly, prokaryotes will just have one circular piece of DNA where we have many linear chromosomes. Okay, so that wraps up this part of the chapter.